Fine, let's start. Number 1. Variola major in Siberian smallpox permafrost graveyards. Along the Kolyma and Lena river systems in Yakutia, the banks are thawing. Seasonal erosion exposes what permafrost kept hidden for generations: animal bones, organic layers, and, in some stretches, human graves. These are not prehistoric burial mounds. Many are epidemic cemeteries from the 18th and 19th centuries, when smallpox moved through Siberian communities with very little resistance. The question researchers now ask is whether the virus itself survived the interval. Variola major is one of the most historically consequential pathogens ever documented. Before global eradication in 1980, it killed an estimated 300 to 500 million people in the 20th century alone. Its mortality rate historically reached roughly 30%. It was declared eradicated after a worldwide vaccination campaign coordinated by the World Health Organization. The last known natural case occurred in Somalia in 1977. That eradication means no population younger than roughly 45 has routine immunity. A new case anywhere on Earth would be a Category A public health emergency. In Siberia, the geography creates a specific convergence of risk factors. Yakutia holds some of the world's deepest and most continuous permafrost. Epidemic cemeteries from past smallpox outbreaks exist in that region. Shallow riverside graves are particularly vulnerable to erosion. Some burial sites were mapped only in local records or remembered by communities, rather than archived in accessible databases. Variola is an orthopox virus. It carries a large double-stranded DNA genome, roughly 185 kilobase pairs. DNA is chemically more stable than RNA, which gives it a theoretical preservation advantage. However, long-term survival still requires specific conditions: stable cold, low oxygen, minimal microbial competition, no repeated freeze-thaw cycling, and protection from ultraviolet radiation. Permafrost can provide most of those conditions in deep, undisturbed layers. The active surface layer, which thaws every summer and refreezes every winter, is far more destructive. Ancient DNA researchers have recovered variola-related genetic sequences from preserved Siberian remains. Those recoveries do not represent living virus. What the studies produced were DNA fragments, enough to reconstruct portions of the viral genome and trace the evolutionary history of smallpox outbreaks across centuries. Researchers, including those working with the Ancient Pathogen Group at the University of Copenhagen and collaborating institutions, have used these fragments to map how variola spread through northern Eurasian populations over time. The molecular record is real. Confirmed infectious particles are not. A virus can leave a molecular fingerprint long after it can no longer infect a cell. Degraded DNA may read clearly on a sequencer while the actual virion is physically destroyed. Envelope proteins, capsid structures, and the structural integrity needed for cell entry can be ruined by ice crystal damage, oxidation, or microbial enzyme activity, even when the genome sequence remains partially readable. There are 3 frameworks for interpreting the Yakutian smallpox situation. The mainstream scientific view holds that the graves preserve degraded genetic material only, and that the probability of encountering infectious smallpox from a thawing cemetery is extremely low. The archaeological and historical interpretation focuses on exceptional preservation. Some grave sites include dried scabs, textile wrappings, and tightly sealed frozen tissue that might retain structural integrity longer than standard decomposition models predict. The speculative framework asks whether rare combinations of cold, darkness, dryness, and burial depth could preserve viable virus in isolated cases. That last scenario has no confirmed example in the smallpox literature, but it shapes how some biosecurity analysts think about worst-case planning. What the graves most reliably preserve is not a living virus. They preserve the record of catastrophes we stopped mapping when we thought the disease was finished. And the next section will show what happens when that assumption holds for a livestock disease, just as dangerous in a different way. Number 2. Brucella suis in Soviet-era frozen reindeer burials, Yamal. The Yamal autonomous okrug is one of the world's largest reindeer herding regions. During the Soviet period, veterinary infrastructure in the area operated through collective management systems. When disease outbreaks struck reindeer herds, the standard response often involved mass culling, followed by burial of carcasses in disposal pits. Those pits were sometimes dug into permafrost, logged in local Soviet veterinary records, and then effectively forgotten after the Soviet administrative system collapsed. Brucellosis in this region is most closely associated with Brucella suis biovar 4, a strain tied specifically to Arctic caribou and reindeer across Eurasia and North America. The disease is enzootic in wild reindeer populations across northern Russia and has historically moved between wild herds and domestic animals when their ranges overlap. Brucella is a gram-negative facultative intracellular coccobacillus. That classification matters for understanding its survival behavior. Unlike Bacillus anthracis, Brucella does not form durable spores. It survives by hiding inside host immune cells, specifically macrophages, which allows it to evade standard immune defenses during active infection. Outside a host, it is less durable than anthrax, but more persistent than many fragile respiratory pathogens. Frozen tissue can protect it from the enzymatic breakdown that would occur in warm, moist soil. The materials that carry the highest Brucella load are reproductive in nature: placenta, aborted fetuses, amniotic fluid, vaginal discharge, and birth-associated fluids. Blood, bone marrow, and raw milk also carry significant bacterial concentrations. In infected reindeer herds, the disease presents as abortions in late pregnancy, weak calves, infertility, retained placenta, and reduced herd productivity over time. These signs are often attributed to nutritional stress or rough terrain conditions, which contributes to underreporting and delayed recognition. In humans, Brucella suis biovar 4 causes what is often called undulant fever, a condition characterized by recurrent fever spikes, night sweats, severe fatigue, and musculoskeletal pain. Joint infection, including vertebral osteomyelitis, can develop in untreated cases and leads to long-term disability. Chronic relapse is a documented feature of human brucellosis and can persist for months or years after initial infection. Veterinary surveillance in the Yamal and Yamalo-Nenets regions has tracked active brucellosis in living herds as the primary modern risk. Thaw, flooding, and thermokarst collapse, which is the ground subsidence caused by melting ice-rich permafrost, can expose old burial pits. Scavengers complicate the picture considerably. Arctic foxes, wolves, ravens, and herding dogs can access thawed carcass material and distribute contaminated tissue across large areas. A dog that scavenges from a thawing Soviet-era burial pit can carry infective tissue directly into a herding camp. The mainstream scientific interpretation is straightforward. Current Brucella risk in Yamalo-Nenets comes primarily from infected living animals, not from ancient frozen remains. The historical interpretation notes that Soviet-era mass burials represent a poorly characterized archive of frozen infected tissue, some of which may have remained biologically intact in cold, stable permafrost. The speculative model considers whether a network of thawing carcass deposits could create reconnected transmission chains between old buried material and active herds moving across the same landscape. Brucellosis will not announce itself with mass die-offs or dramatic outbreak signals. It works as a slow fever that moves through meat, milk, and handling practices. The next case is faster moving, and it begins with something as small as a dead vole under a snowbank. Number 3. Frozen Francisella tularensis in Arctic rodent carcasses. Rodent populations in Arctic and subarctic ecosystems move in cycles. Lemming and vole populations build over several years, then crash. During a population peak, animals are dense on the landscape. During a crash, carcasses accumulate in large numbers across the tundra, snow surface, and shallow burrow systems. Those carcasses do not disappear instantly. In cold conditions, they freeze and persist through the winter. When spring arrives and the snowpack melts, the material they contain enters the water system. Francisella tularensis is the bacterium responsible for tularemia. It is classified as a highly infectious, gram-negative intracellular organism. The infective dose for human exposure through inhalation has been estimated at as few as 10 to 50 organisms. That exceptionally low threshold makes carcass contamination in water or soil a meaningful exposure risk, even at low concentrations. Tularemia has a documented history in Russia, Scandinavia, and North America as an occupational hazard for hunters, trappers, and water workers. Transmission routes are multiple. Skin abrasions can allow entry when handling infected animals or carcasses. Eye exposure, known as oculoglandular tularemia, occurs through splashed contaminated fluid. Ingestion of contaminated water causes oropharyngeal tularemia. Tick and deer fly bites transmit the bacterium through arthropod feeding. An inhalation of contaminated aerosols or dust produces pneumonic tularemia, the most clinically severe form, which can progress rapidly to respiratory failure without prompt antibiotic treatment. Frozen carcasses matter in this disease system because tissue insulates and protects bacteria during winter. When meltwater moves through a spring thaw event, it can carry bacteria from multiple carcass sites into ponds, wetland channels, and drinking water sources downstream. The bacterium has been recovered from environmental water sources in Scandinavia during outbreaks that followed the pattern of rodent die-offs with subsequent snowmelt. Arctic foxes are ecologically important sentinels in this system. They feed extensively on rodents and scavenge carcasses throughout the year. When tularemia is circulating in a rodent population, foxes accumulate antibody titers that researchers can detect through blood sampling. Wildlife biologists working in Russia, Norway, and Alaska have used fox serology as a proxy for landscape-level tularemia activity in areas too remote for direct rodent surveillance. Climate warming intersects with this system in several ways. Longer warm seasons change rodent reproductive cycles and potentially compress the time between boom and crash phases. Altered precipitation patterns, including more frequent rain-on-snow events that form ice crusts on the tundra surface, can disrupt lemming burrow access and accelerate die-offs. Expanded tick and deerfly ranges push the arthropod transmission route into higher latitudes than it historically occupied. The mainstream interpretation treats tularemia as an ecologically persistent disease, whose risk is driven by modern rodent and arthropod populations. The historical reading focuses on frozen carcass banks as wintertime bacterial reservoirs that release their contents during spring melt. The speculative scenario considers whether older frozen rodent remains, buried under permafrost rather than just seasonal snowpack, could seed isolated water systems during deep thaw events. A disease does not need to emerge from a mammoth to be a permafrost-linked risk. Sometimes it only needs a vole under snow. What follows takes that same logic and applies it to something that can travel without any animal contact at all, just dried dust on the wind. Number 4. Q fever in Soviet-era livestock burials. Coxiella burnetii, the bacterium that causes Q fever, is classified as a biosafety level 3 pathogen. Its most distinctive characteristic is environmental durability. Coxiella produces a small cell variant, a condensed cellular form that is not a classical endospore the way anthrax is, but functions with comparable resistance to drying, heat, and chemical disinfectants. It can remain viable in dry soil, dust, and contaminated wool or hides for months to years under ambient outdoor conditions. Soviet-era livestock operations in Arctic and subarctic Russia maintained large herds of sheep, goats, and cattle in collective systems. When animals died in large numbers from disease, starvation, or planned culling campaigns, Disposal often involved burial in pits dug into available ground. In permafrost regions, those pits would have been shallow, which means material was placed at or near the active layer, rather than in stable frozen ground. Veterinary documentation from those sites may be held in regional Soviet archives, many of which remain poorly digitized or physically inaccessible. The highest risk biological materials for Q fever are birth-associated. A single parturition event in an infected sheep or goat can release up to 1 billion organisms per gram of placenta. Contaminated placenta, amniotic fluid, aborted fetuses, and birth bedding are the primary vectors. Urine, feces, and milk also carry the bacterium. In a buried disposal site, these materials may remain frozen and biologically intact within stable permafrost. Thaw begins the problem, but drying completes it. The aerosol transmission route is the critical mechanism for human disease at distance. When contaminated birth waste or infected soil dries after thaw, particles become airborne. Wind can carry them beyond the burial site perimeter. People downwind from a drying burial pit, road cut through contaminated soil, or vehicle-disturbed burial mound can inhale infective particles without any direct contact with animals. Human Q fever begins with acute symptoms that look like influenza: fever, intense headache, chills, and fatigue. Pneumonia and hepatitis can develop in more severe cases. The most serious long-term complication is chronic Q fever, primarily in the form of endocarditis or vascular graft infection, especially in people with pre-existing cardiac valve disease or immune compromise. Chronic Q fever carries significant mortality if untreated. Veterinary epidemiology and environmental sampling form the research basis for Q fever surveillance in livestock regions. Serological surveys of sheep and goat herds in Russia have documented Coxiella exposure in multiple regions. Outbreak reconstruction has traced human cases to specific lambing seasons, slaughterhouse operations, and contaminated agricultural dust events. The Netherlands experienced a large Q fever epidemic between 2007 and 2010, primarily linked to dairy goat operations, which gave researchers substantial modern epidemiological detail about exposure and transmission at population scale. The mainstream scientific view is that active livestock herds are the dominant source of Q fever exposure. The historical interpretation holds that frozen Soviet disposal pits may preserve birth-associated materials in a state that thaw, and drying could reactivate as a dust source decades later. The speculative framework imagines a cycle where permafrost thaw, summer drying, and wind work together in a seasonal pattern, turning old pits into recurring aerosol sources rather than a single release event. The distinguishing feature of this pathogen is that it does not need to bleed or flow. It only needs to dry. What comes next is not a frozen pathogen at all, but a disease that is advancing for a different reason, one that makes it a useful bridge between the thaw stories and the broader climate picture. Number 5. Tick-borne encephalitis virus expanding out of thawing Siberian permafrost. The title of this case requires an immediate clarification. Tick-borne encephalitis virus is not thawing out of frozen ground. It has no established reservoir in permafrost. The disease is spreading because its vector is advancing, and the reason the vector is advancing is directly connected to the same warming process, reshaping the permafrost landscape. That distinction matters for accuracy, and it matters for understanding how climate-linked disease works. Tick-borne encephalitis, or TBE, is caused by a flavivirus in the same family as dengue, Japanese encephalitis, and West Nile virus. In Eurasia, the primary vectors are Ixodes ricinus in the west and Ixodes persulcatus in Siberia and the Russian Far East. The virus is maintained in a cycle between ticks and small mammals, primarily rodents, which serve as reservoir hosts without developing clinical disease. Ticks pick up the virus during larval or nymphal blood meals on infected rodents and transmit it during later feeding stages on larger hosts including reindeer, livestock, and humans. The tick life cycle has specific environmental requirements. Larvae, nymphs, and adults each need blood meals, and each needs sufficient humidity to avoid desiccation between feeding stages. Cold winter temperatures traditionally constrained how far north these ticks could survive and reproduce. As Siberian winters have warmed and become shorter, the zone of survivable tick habitat has shifted northward by measurable distances. Snow insulation during mild winters can protect tick populations at the soil surface from lethal cold snaps. Longer summer seasons provide more feeding opportunity and faster larval development. Vegetation change compounds the habitat shift. Shrub expansion into previously open tundra zones, sometimes called shrubification, creates physical cover that traps moisture and moderates surface temperatures. Ixod's ticks require shaded, humid, ground-level environments. As shrub cover extends north and up hillsides in Siberia, it creates tick-compatible microhabitats in areas that were previously inhospitable. Birds are an underappreciated transport mechanism. Long-distance migratory birds can carry infected ticks from established TBE zones into newly colonized northern areas during spring migration. This can seed tick populations in locations ahead of the main geographic front of habitat expansion, creating isolated clusters of infection risk before the connecting corridor of suitable habitat develops. Human TBE illness frequently follows a biphasic pattern. The first phase is a nonspecific febrile illness resembling influenza, lasting approximately a week, followed by a brief apparent recovery. In a significant proportion of patients, a second phase then develops involving the central nervous system, meningitis, encephalitis, or combined meningoencephalitis. Severe outcomes include motor paralysis, cognitive impairment, and long-term neurological sequelae. Case fatality rates vary by viral subtype. The Siberian subtype carries higher mortality and more chronic neurological complications than the European subtype. Raw or unpasteurized milk from infected goats, sheep, and cattle represents an alternative exposure route that public health authorities in Russia and Central Europe have documented in cluster outbreaks not associated with tick bites. Climate modelers and tick ecologists, including research groups at the Russian Academy of Sciences and collaborating European institutions, have mapped range expansion using temperature thresholds, host abundance models, and reported case data. Northern case reports in areas with no previous TBE history have appeared in medical literature with increasing frequency since the 1990s. The mainstream scientific view identifies habitat expansion driven by warming as the primary engine of TBE spread. The historical interpretation notes that land use change, pipeline construction, military activity, and forestry bring people into newly colonized tick zones independently of temperature. The speculative framework, which has minimal supporting evidence, raises the question of whether thawing animal remains could play any indirect role in local viral maintenance. But researchers working in this field consistently identify living ticks and rodents, not ancient ice, as the relevant transmission system. The virus is not waking from the ice. The ticks are marching north. And if you want to see what actually came out of the ice and killed thousands of animals, the next case provides the answer. Number 6. The Yamal anthrax resurrection. In the summer of 2016, the Yamal Peninsula recorded its highest temperatures in roughly 75 years. The active layer of permafrost thawed more deeply than normal. Somewhere in the landscape, possibly beneath a frozen reindeer carcass or near an old burial focus from a previous anthrax outbreak, a population of bacterial spores entered the soil, vegetation, and meltwater of the active grazing land. More than 2,000 reindeer died. Approximately 90 people were hospitalized. One child died. The Yamal anthrax outbreak was not theoretical. It was documented, investigated, and confirmed. Anthrax is caused by Bacillus anthracis, a gram-positive, spore-forming bacterium. The spore is the central feature of its persistence. When conditions become unfavorable for vegetative bacterial growth, Bacillus anthracis forms an endospore, a metabolically dormant, structurally reinforced survival unit enclosed in a multi-layered protein coat. Anthrax spores are among the most environmentally durable biological structures known. They have been recovered and found viable from soil samples decades after the original contamination. In cold, stable permafrost, the theoretical survival window is considerably longer. The mechanism in Yamal followed a sequence that researchers had modeled before the outbreak occurred. Unusually warm temperatures drove active layer thaw beyond the depth that previous warm summers had reached. The thaw exposed old contaminated material, most likely a carcass or burial site from a previous anthrax outbreak in the region, possibly from the early 20th century, when anthrax was common in Yamal reindeer herds. Spores entered soil and surface water. Grazing reindeer ingested spores while feeding on vegetation and drinking from meltwater pools. Once infected animals died, their carcasses amplified contamination. Anthrax bacteria grow vegetatively in fresh carcass blood and tissue, releasing billions of new spores as the carcass is exposed to air. This amplification phase made the contaminated zone substantially larger than the initial thaw focus. Some reports recorded approximately 2,650 infected reindeer and about 2,350 deaths from the outbreak. Human exposure in Yamal occurred primarily through carcass handling, during the emergency response, and through contaminated meat. Anthrax presents in four main clinical forms: cutaneous, gastrointestinal, inhalational, and injection-associated. In the Yamal event, gastrointestinal anthrax was the primary concern for humans because infected meat had entered consumption within herding camps before the outbreak was recognized. Historical anthrax foci in the Russian Arctic are documented but incompletely mapped. Old burial sites from the 1910 to 1940 period, when large anthrax die-offs occurred in Yamal reindeer populations, were not GPS tagged. Many are known through local oral tradition among Nenets herders or through incomplete veterinary records. The combination of forgotten burial sites and poor documentation created a landscape where the biological hazard was present, but its location was uncertain. Vaccination gaps compounded the risk. Routine anthrax vaccination programs for reindeer herds in the Yamal region had been inconsistent in the years before 2016. When the outbreak began, a large portion of the reindeer population lacked protective immunity. Climate scientists, epidemiologists, and veterinary researchers who investigated the outbreak, including teams from Rospotrebnadzor and the Russian scientific community working in the Yamalo-Nenets region, concluded that the heat-driven thaw mechanism was the triggering event. The historical interpretation emphasizes that the buried foci created during previous outbreaks were the stored biological material that the thaw released, not some ancient reservoir. The speculative extension of the Yamal case asks what would happen if multiple buried foci thawed simultaneously during an extended heat wave across a large portion of the peninsula. Yamal proved that permafrost is not only a freezer, it is an archive of unfinished outbreaks. That proof shifts the discussion from possibility to documented reality, and the cases that follow take us somewhere different. Not out to the tundra, but deep underground, where something has been working on chemistry problems for 4 million years. Number 7. Lechuguilla Caves, 4 million-year-old superbugs. Lechuguilla Cave in New Mexico is not a permafrost site. It is not in the Arctic. It does not contain a frozen pathogen. It belongs in this investigation because it answers a question that the previous cases raise. If microbes have been isolated from all modern human activity for millions of years, what do they know about chemical warfare that we do not? Lechuguilla was accessed by researchers for the first time in 1986 when cavers pushed through a previously sealed passage. The main cave system extends more than 240 kilometers and reaches depths of over 480 meters. Its microbial communities have evolved in complete isolation from the surface world, from hospitals, from agriculture, from any antibiotic compound produced by modern pharmaceutical industry. In 2012, researchers led by Jerry Wright at McMaster University published a study on bacteria isolated from Lechuguilla. They found that many cave bacteria carried resistance to commercially produced antibiotics, in some cases showing resistance to as many as 14 different drug classes simultaneously. These were not bacteria that had been exposed to medical antibiotics and evolved resistance in response. They had never encountered those compounds. Their resistance mechanisms were ancient: drug-destroying enzymes, modified drug-binding sites, altered membrane permeability, and active efflux pumps that eject toxic compounds from bacterial cells before damage occurs. The study introduced the concept of the resistem in a clarifying context. The resistem is the complete set of antibiotic resistance genes present in a microbial community. Wright's work demonstrated that the resistem is not a modern artifact of pharmaceutical misuse. It is a feature of microbial biology that predates human civilization by an enormous margin. Bacteria have been chemically competing with each other for hundreds of millions of years. The compounds they produce to kill competitors are, in many cases, the same classes of compounds that pharmaceutical scientists later identified and developed into antibiotics. The resistance mechanisms evolved in parallel with those compounds in bacterial communities that never interacted with a hospital. Horizontal gene transfer is the mechanism that makes this relevant beyond the cave walls. Bacteria can transfer genetic material between individual cells and even between distantly related species through plasmids, transposons, and bacteriophages. A resistance gene that evolved in a cave bacterium 4 million years ago is not permanently confined to that lineage. If those bacteria contact modern pathogens, genetic exchange is possible under the right conditions. The Lechuguilla findings do not describe confirmed human pathogens. The cave isolates are not causing clinical infections, but their genes represent a library of resistance mechanisms that modern medicine has not fully cataloged. The mainstream interpretation is precise. Ancient resistance is a natural feature of microbiology, but clinical danger requires those genes to transfer into pathogens capable of infecting humans and causing disease in modern settings. The historical interpretation notes that isolated ecosystems, caves, ice pockets, deep sediments, and permafrost cores preserve pre-human microbial strategies that the era of antibiotic medicine never encountered during development. The speculative extension imagines that other sealed environments, including Arctic permafrost in deep stable layers, might harbor microbial communities with comparable resistance profiles and unknown metabolic capabilities. Lechuguilla did not preserve a plague. It preserved the instruction manual for surviving our drugs. The next case takes us from a cave in New Mexico back to frozen ground, but this time with a pathogen whose genome was not just found in permafrost. It was recovered, sequenced, and reconstructed. Number 8. The 1918 influenza virus preserved in Alaskan permafrost. In November of 1918, Breivik Mission was a small Inuit community on the Seward Peninsula of Alaska. The influenza pandemic reached the village within a matter of days. Of roughly 80 adult inhabitants, 72 died. Survivors buried the dead in a mass grave. Permafrost beneath the site preserved the bodies in cold storage for the following 8 decades. The 1918 H1N1 pandemic, sometimes called the Spanish flu, killed an estimated 50 million people worldwide, though some more recent demographic estimates have placed the toll closer to 100 million. It is the single most lethal infectious disease event in recorded human history. What made it biologically unusual was the age distribution of mortality. Most influenza outbreaks cause disproportionate death in the elderly and in infants. The 1918 virus killed in a W-shaped mortality curve, meaning it caused unusually high mortality in young adults between approximately 15 and 34 years old. The biological mechanism behind that pattern was not understood for most of the 20th century. Influenza is an enveloped RNA virus. RNA degrades significantly faster than DNA under most environmental conditions. The lipid envelope that surrounds the influenza virion is disrupted by freeze-thaw cycling, oxidation, and microbial enzyme activity. Standard virology predicted that recovering intact, sequenceable RNA from an 80-year-old frozen human corpse would be extremely difficult. Johann Holton, a pathologist who had been thinking about the Breivik mission graves since the 1950s, returned to the site in 1997 with permission from community elders and the village council. He recovered lung tissue from one preserved individual. That tissue contained enough viral RNA fragments for a research group at the Armed Forces Institute of Pathology, led by Jeffrey Taubenberger, to sequence portions of the 1918 influenza genome. The project eventually produced a complete genomic sequence of the virus from Alaskan permafrost material and other archived samples from the period. The sequencing work revealed specific features of the 1918 virus that explained its unusual virulence. Its hemagglutinin gene showed adaptation features consistent with avian influenza origins. The polymerase complex contained mutations associated with enhanced replication in human cells. Comparisons with later pandemic strains, including the 1957 H2N2 and the 1968 H3N2 viruses, clarified the evolutionary trajectory of pandemic influenza in the 20th century. In 2005, researchers at the Centers for Disease Control and Prevention, working with the reconstructed genome sequence, used reverse genetics to recreate the 1918 virus in a biosafety level 4 laboratory. That work produced significant scientific insight and significant controversy. The primary scientific finding was that the virus could infect multiple mammalian hosts efficiently and caused severe disease in animal models. The controversy centered on biosecurity. Whether reconstructing a pandemic-capable virus from genetic sequence information created risks that outweighed the research benefits. The modern pandemic influenza threat comes from reassortment events in birds and pigs, not from natural thawing of permafrost graves. The Alaskan site does not pose an ongoing infectious release risk. The virus recovered there required deliberate laboratory reconstruction. The mainstream interpretation is that permafrost preserved genetic material sufficient for scientific analysis, but not naturally infectious virions. The historical interpretation notes that frozen human graves from epidemic periods may preserve enough molecular evidence to reconstruct lost outbreak histories, which has direct application to surveillance for unknown historical pandemics. The speculative interpretation considers whether other mass graves from undocumented epidemic events in Arctic or subarctic regions might contain similarly preserved genomic material from pathogens not yet identified. The virus did not crawl out of the grave. Science entered the grave and recovered its blueprint. What comes next proves that the ice can preserve not just a blueprint, but a working virus, though the host it infects is not a human being. Number 9. Pithovirus sibericum. In 2014, a team led by Jean-Michel Claverie and Chantal Abergel at Aix-Marseille University published a paper in the Proceedings of the National Academy of Sciences describing the revival of a functional virus from Siberian permafrost, more than 30,000 years old. The organism was named Pithovirus sibericum. Its host is amoebas of the genus Acanthamoeba. It does not infect human cells. That last point is critical and should not be minimized. The significance of Pithovirus is not that it threatens human health directly. Its significance is what it demonstrates about long-term viral viability in permafrost conditions. Pithovirus belongs to a group called giant viruses, a category of large DNA viruses discovered primarily in the last 2 decades. Its particle is elongated, approximately 1.5 micrometers in length, which is visible under a standard light microscope and approaches the size of small bacteria. This physical scale is extraordinary by virology standards. Most human pathogenic viruses range from about 80 to 300 nanometers. Pithovirus is roughly 5 to 15 times larger in linear dimension than common respiratory viruses. The genome of Pithovirus, despite the enormous physical size of the particle, is relatively modest by giant virus standards, approximately 610,000 base pairs. Much of the particle interior is not nucleic acid, but appears to be structural matrix. The particle architecture may contribute to its exceptional preservation by physically protecting the genome from environmental damage. The revival experiment used Acanthamoeba castellani cells as host. Researchers exposed the cells to thawed permafrost material and observed the production of new viral particles, which confirmed that the virus retained the ability to complete its replication cycle after more than 30,000 years in frozen storage. This was not sequencing. This was not detection of DNA fragments. This was demonstrated infectivity. Giant viruses use amoeba hosts in ways that have implications beyond simple infection. Amoebas are microbial mixing chambers. They ingest bacteria, viruses, and other microorganisms from the environment. Within amoeba cells, organisms from different lineages can interact, exchange genetic material, and potentially recombine. Researchers studying giant virus ecology, including members of Claverie and Abergele's group, and independent teams at other institutions, have characterized amoeba hosts as potential hotspots for horizontal gene transfer and genetic mixing between distantly related organisms. The mainstream interpretation treats Pithovirus as a proof of principle for ancient amoeba virus survival in permafrost. It does not suggest that human pathogenic viruses survive under comparable conditions, because the biological and structural requirements differ substantially. The historical interpretation notes that permafrost environments preserve whole microbial ecosystems, not individual curiosities. The samples that yielded Pithovirus also contained bacteria, fungi, and other viral sequences from multiple lineages. The speculative interpretation asks whether human or animal pathogens, particularly non-enveloped DNA viruses with stable capsid structures, could also retain viability under rare, optimal permafrost preservation conditions. No confirmed example exists, but the biology does not make it impossible. Industrial expansion in the Arctic, including mining operations, pipeline construction, drilling programs, and mammoth tusk hunting, which disturbs deep permafrost layers in pursuit of ivory, can expose ground that has been sealed since before the last glacial maximum. Most of what they release will be microorganisms with no human hosts. Pithovirus is not the monster. It is the warning flare. And when the ground itself tears open, as it now literally does in one corner of western Siberia, the exposure question moves from the gradual to the sudden. Number 10. The Siberian Crater Plague Factories. In July of 2014, a helicopter survey team flying over the Yamal Peninsula photographed a circular collapse feature in the tundra, a pit roughly 30 meters across, with steep walls exposing raw permafrost and a visible opening to depth. It was the first of what would become a recognized category of Arctic geological event. Similar features subsequently appeared in the Gaiden Peninsula and elsewhere in the Yamal region. By 2020, researchers had cataloged dozens of these structures from satellite and aerial imagery. The term plague factory is a metaphor and not a formal scientific designation. These craters are not confirmed pathogen vents. They are not meteor impacts or volcanic features. They are the product of subsurface pressure dynamics in warming permafrost terrain. The formation mechanism, as reconstructed by geologists, including teams at the Skolkovo Institute of Science and Technology and the Arctic and Antarctic Research Institute in St. Petersburg, involves a combination of factors. Ice-rich permafrost thaws from above and below. Methane gas, produced by decomposing organic matter in the subsurface, and potentially destabilized gas hydrates, accumulates beneath a weakening frozen cap. Saline water bodies, called cryopegs, which remain liquid year-round within permafrost due to high salt content, may also contribute hydraulic pressure. When the frozen cap fails, the ground either collapses inward or ejects material outward, creating a crater with exposed permafrost walls. Those walls continue thawing after formation. Meltwater, soil particles, organic debris, and any biological material present in the exposed permafrost layers enter the drainage system. Over time, many craters fill with water and become thermokarst lakes, which preserve and redistribute organic material from ancient layers across the surrounding landscape. Animals are drawn to new water sources. Reindeer, foxes, birds, and insects congregate at crater lakes. If those water bodies contain biological material from old carcass deposits or infected soil layers, the crater has created a new contact zone where multiple species interact with formerly sealed material. The confirmed analogy is anthrax. The Yamal crater features occur in the same region where the 2016 anthrax outbreak took place. Old anthrax burial foci are distributed across Yamal in locations that are incompletely mapped. Ground collapse in the right location could expose spore-bearing soil layers that would otherwise remain inaccessible. Contaminated meltwater, grazing reindeer, and scavenging animals would complete the transmission chain. Atmospheric scientists studying methane release from Arctic permafrost, including groups led by researchers such as Natalia Shakova at the University of Alaska Fairbanks and Sergei Zimov at the Northeast Science Station in Chersky, Russia, have noted that revived microbial communities in thawing organic matter contribute to carbon and methane release as a climate feedback. The same microbial reactivation that drives greenhouse gas output from permafrost also confirms that biological material in those layers was preserved in a metabolically recoverable state. The mainstream interpretation treats craters as geological and climate features with indirect disease risk. They can expose buried material, but confirmed pathogen emergence from craters has not been documented beyond the general anthrax geography. The historical interpretation focuses on the unknown archaeology of the buried terrain, which predates modern mapping and could contain carcass deposits, burial grounds, or epidemic foci that have no recorded location. The speculative interpretation raises the possibility of unknown ancient microbial or viral communities in the exposed permafrost walls, a hypothesis with no current supporting evidence, but one that researchers studying permafrost viromes consider worth monitoring. Craters do not create plagues. They open the locked rooms where old outbreaks were buried. What determines whether those rooms contain something dangerous, and how we would know, requires understanding the physics of why anything survives in frozen ground at all. Permafrost is not magic. It preserves biological material by slowing the chemical processes that normally destroy it. Enzymatic decomposition, oxidative damage, microbial predation, and temperature-driven molecular degradation. Understanding those processes in physical terms explains why some organisms survive and why others do not. Permafrost is defined as ground that remains frozen for at least 2 consecutive years. In practice, much of the Siberian permafrost has been continuously frozen for thousands to hundreds of thousands of years. The deepest and oldest layers have been stable since the Pleistocene. Above the permafrost lies the active layer, the zone that thaws each summer and refreezes each winter. The active layer in Siberia typically ranges from 30 centimeters to a few meters in depth, varying with latitude, soil type, vegetation cover, and recent temperature trends. As temperatures warm, the active layer deepens. Deeper active layer thaw means reaching older biological material. Temperature stability is more important than simply being cold. Repeated freeze-thaw cycling is destructive to cellular structures. Ice crystals that form during freezing expand inside cells and viral particles, rupturing membranes and envelopes. An organism that freezes and thaws once may survive. An organism that cycles through that process dozens or hundreds of times across seasonal active layer fluctuations is progressively destroyed. Organisms preserved in deep, stable permafrost below the active layer are protected from that cycling. Darkness matters for nucleic acid preservation. Ultraviolet radiation from sunlight destroys DNA and RNA by inducing crosslinks and strand breaks. Permafrost, by definition, is buried from sunlight. Low oxygen content further slows oxidation reactions that degrade lipids, proteins, and nucleic acids. The combination of cold, dark, and anoxic conditions gives permafrost its preservation properties. Different pathogen types have very different survival profiles. Bacterial spores, particularly Bacillus anthracis spores, are the most durable. Their multi-layered protein coat, calcium dipicolinate core, and dehydrated interior provide structural protection that has no equivalent among viruses. Non-spore bacteria can survive if protected inside host tissue, host cells, biofilms, or dense organic matrices. Once exposed to open soil, they degrade much more rapidly. Viruses divide into two broad stability categories. Non-enveloped viruses, which have a protein capsid as their outermost layer and no lipid membrane, are generally more stable than enveloped viruses. Parvoviruses, adenoviruses, and some caliciviruses in this category can survive environmental exposure for extended periods. Enveloped viruses, including influenza, coronaviruses, herpes viruses, and pox viruses like Variola, depend on an intact lipid bilayer for infectivity. That layer is vulnerable to freeze-thaw disruption, oxidation, and detergent-like compounds in soil. DNA viruses with non-enveloped capsids and large, stable genomes, like the giant viruses in the Pithovirus family, are the most likely candidates for very long-term permafrost viability. Carcass tissue acts as insulation. A frozen animal body creates a microenvironment with different moisture, oxygen, and temperature characteristics than surrounding soil. Microbes embedded deep within frozen tissue may be partly shielded from conditions that would destroy organisms in direct contact with the soil matrix. The question is not whether anything can survive in permafrost. Demonstrated survival exists across multiple organism types. The question is, what survived, in what condition, at what depth, in what burial context, and whether any of it retains the ability to infect modern hosts. That is a question science answers case by case. The same thawing ground looks different depending on which scientific discipline is doing the looking. Epidemiologists, archaeologists, climate scientists, and molecular biologists each bring analytical frameworks that emphasize different aspects of the same evidence. Three coherent interpretive positions are worth examining in terms of what each predicts, what evidence it relies on, and where its limits are. The mainstream scientific position starts from established ecology. Anthrax, brucellosis, tularemia, Q fever, and tick-borne encephalitis are not theoretical risks. They are documented zoonotic diseases with known distribution in Arctic and subarctic environments. They cause real infections in real people in the present day. The mainstream argument is that climate warming intensifies these existing risks through predictable mechanisms. Deeper thaw exposes more anthrax spore foci. Warmer summers expand tick ranges. Altered hydrology changes, tularemia transmission in meltwater systems, and stressed reindeer herds face greater brucellosis burden. The Yamal anthrax outbreak of 2016 is the clearest proof point. Known pathogen, known mechanism, documented outcome. The mainstream position also cautions against amplifying fear about ancient unknown pathogens, when better characterized known pathogens present more immediate and actionable risks. The archaeological and historical interpretation operates from a different archive. It treats permafrost as a stratigraphic record of past epidemic events and notes that human memory, administrative records, and cartography have all failed to document much of what was buried. Soviet veterinary burial sites, indigenous epidemic cemeteries, expedition graves, and wartime animal disposal pits exist in quantities that formal science has not cataloged. The historical position does not primarily fear unknown monsters. It fears familiar pathogens in forgotten locations. Old brucellosis burial pits, old anthrax foci, old Q fever sheep burial grounds, and old smallpox epidemic cemeteries are all potentially preserved in permafrost terrain that is now actively thawing, and in many cases being physically disturbed by infrastructure development. The historical interpretation treats archival work, oral history, and burial site mapping as legitimate scientific tools alongside field sampling and laboratory analysis. The fringe and speculative interpretation uses Pithovirus as its primary proof of principle. If a 30,000-year-old amoeba virus can survive in frozen Siberian ground and infect its host upon revival, the boundary conditions for ancient pathogen persistence are wider than standard preservation models would predict. The speculative framework asks about deep permafrost viromes, cryopeg microbial communities, sealed Arctic cave ecosystems analogous to Lechuguilla, and the large fraction of permafrost microbial diversity that has not yet been characterized by metagenomic sequencing. It takes seriously the possibility that human or animal pathogens of ancient origin could retain some degree of viability under rare optimal conditions. The weakness of this position is host compatibility. A virus that evolved in a mammal population that no longer exists may be unable to infect modern immune systems efficiently, and ancient envelope proteins may not bind current cell surface receptors. The intellectually honest position treats all three frameworks as operating on different timescales and different types of evidence. Mainstream analysis points to near-term documented risk. Historical analysis points to recoverable but currently hidden risk. Speculative analysis points to low probability but high consequence scenarios that are worth monitoring precisely because their probability is not zero. Thaw is reopening biological history at uneven rates across the Arctic landscape. The scientific community does not yet have a complete map of what it contains. The physical process of detecting a pathogen emergence in a remote permafrost landscape involves layered tools operating at different scales, from satellite imagery to individual blood samples. The outer layer of detection is geographic. Remote sensing platforms track land surface temperature, active layer depth, and ground collapse features across the Siberian landscape. Satellite-based synthetic aperture radar can identify thermokarst formation, crater development, and permafrost subsidence over large areas without requiring ground access. Drone surveys extend resolution into areas identified by satellite as actively changing. Thermal imaging maps areas where ground temperature anomalies suggest accelerated thaw. Veterinary surveillance forms the second layer. Sudden, unexplained herbivore mortality is the primary trigger for field response in anthrax risk areas. Veterinary teams in the Yamal and Yamalo-Nenets regions maintain protocols for reporting mass reindeer deaths, which can initiate rapid deployment of field investigators and laboratory sampling. Blood samples from herding dogs, which may scavenge infected carcasses, serve as sentinels for anthrax, brucellosis, and tularemia. Fox serology, as discussed in the tularemia section, provides a landscape-scale proxy for rodent-borne disease activity. Tick drag sampling, using flannel sheets dragged across vegetation transects, measures tick density and geographic distribution for TBE surveillance. Environmental sampling operates at the molecular level. Metagenomics, the direct sequencing of all nucleic acids in an environmental sample, allows detection of microbial diversity without the need to culture organisms in a laboratory. Permafrost cores, meltwater samples, and sediment from thermokarst lakes can all be analyzed by metagenomics to identify viral, bacterial, and fungal sequences, including sequences from organisms that cannot be grown in standard laboratory conditions. Environmental DNA detection can establish whether an organism is present in a sample, but cannot by itself confirm whether that organism is viable and infectious. Viability testing requires different methods. Culture-based revival, placing sample material in contact with appropriate host cells or growth media under biosafety containment, is the only definitive demonstration of infectivity. For potentially dangerous pathogens, this work requires biosafety level 3 or 4 containment. The scientific community has active debates about when such revival experiments are scientifically justified, and whether the knowledge gained outweighs the biosafety risk of confirming that a dangerous ancient organism remains infectious. Public health detection in the field depends on clinical recognition. Remote Arctic clinics serve populations with limited access to specialist diagnostics. A herder presenting with cutaneous ulcer and fever may have cutaneous anthrax or tularemia, both of which can be mistaken for bacterial wound infection. A child with biphasic febrile illness and developing neurological signs may have TBE, which requires specific serology to confirm. Diagnostic capacity in remote Siberian, Alaskan, and northern Canadian communities is often insufficient for rapid identification of rare diseases. Indigenous reindeer herders, hunters, and community members represent a first detection system that exists independently of formal surveillance infrastructure. They observe herd behavior, recognize unusual carcass distribution, notice changes in water sources, and identify unexplained illness in community members earlier than any remote monitoring system. Integrating that observational knowledge with formal surveillance programs has been consistently identified by Arctic disease researchers, including those working with the Arctic Council's programs and with circumpolar health research networks, as a critical gap in current outbreak detection capacity. Detection is the difference between a strange carcass on the tundra and a regional emergency, and the people who first encounter that carcass are the subject of the next section. The gradient of exposure to permafrost-linked disease runs from people whose lives are entirely organized around the thawing landscape to people who enter it for industrial purposes and leave. Understanding who faces what risk requires specificity. Nomadic reindeer herders in Yamal, the Sakha Republic, and the Chukotka Autonomous Okrug are the population group with the most consistent and comprehensive exposure. Their migration routes cross landscapes where old carcass deposits, anthrax burial foci, and Soviet-era disposal pits exist. They handle carcasses during slaughter, prepare raw organs and marrow as food, dress hides, and may access water from streams that drain thawing terrain. In an anthrax event, herders who slaughter infected animals or consume infected meat can develop cutaneous or gastrointestinal disease before any official response has begun. The Yamal 2016 event demonstrated exactly that sequence. Hunters and trappers face tularemia risk through direct contact with hares, rodents, and foxes, through water sources in the field, and through tick and deer fly exposure during warm months. Hunters operating in areas of range expansion for Ixodes ticks encounter TBE risk in zones where the disease may not yet be clinically recognized. A hunter who develops biphasic febrile illness in a remote hunting camp and attributes it to standard flu may not seek medical attention until neurological symptoms develop. Veterinarians and slaughter workers face the highest occupational exposure to brucellosis through reproductive tissues and to Q fever through birth products. The aerosol route for Q fever is particularly difficult to control in open outdoor slaughter settings. Protective equipment is rarely used routinely in traditional subsistence slaughter practices. Industrial workers introduce a different exposure profile. Road construction, pipeline installation, mining, and drilling can disturb deep permafrost in areas with no recent surface thaw. These activities can expose biological material in undisturbed layers that would not normally interact with the surface environment for decades. Mammoth tusk hunters, who excavate permafrost sediments in Siberia in search of ivory, work in conditions very similar to the laboratory permafrost core research that has yielded Pithovirus and ancient bacterial sequences, but without biosafety protocols. Misdiagnosis is a consistent risk across all exposure groups. Anthrax, tularemia, brucellosis, Q fever, and TBE all present initially with fever and malaise that is indistinguishable from common viral illness in the first 24 to 72 hours. Remote clinics in Siberia, Alaska, and northern Canada typically lack point-of-care diagnostic capability for any of these diseases. Laboratory confirmation requires either regional reference, laboratory capacity, or samples transported to central facilities, which can take days. Treatment delay in gastrointestinal anthrax or pneumonic tularemia significantly worsens outcomes. Food security and cultural continuity are inseparable from the biological risk. If a brucellosis outbreak reduces herding productivity or forces culling of infected animals, the economic and subsistence consequences extend across entire communities. Quarantine measures, carcass burning requirements, and movement restrictions imposed during an anthrax response can conflict directly with traditional herding practices and may generate resistance that slows public health response. The first witnesses to a resurfacing disease will not be at a laboratory bench. They will be moving across the thaw line, reading the landscape for signs that something has changed. Permafrost does not create pathogens. It stores them, hides them, and eventually releases whatever it preserved under the right conditions. That distinction controls how every case in this investigation should be understood. The cases divide along a hierarchy of certainty. At the highest certainty level sits anthrax. Bacillus anthracus spores are documented in Siberian permafrost. Thaw exposed them. Reindeer died. People were hospitalized. The chain of events was observed, sampled, and confirmed. The mechanism is not speculative. It occurred in 2016 on the Yamal Peninsula. The next tier contains genetic archive cases. The smallpox DNA fragments recovered from Yakutian remains, and the 1918 influenza genome sequenced from Alaskan permafrost demonstrate that frozen graves can preserve the molecular record of past outbreaks. They do not demonstrate naturally infectious released pathogen, but they demonstrate that the archive function of permafrost is real and scientifically productive. Pithovirus sits alone in its category. It is the only confirmed revival of an ancient virus demonstrating infectivity after genuine geological timescales. Its host is an amoeba. That limitation is scientifically important. But viability over 30,000 years of permafrost storage has been demonstrated, not inferred. Lechuguilla contributes a different lesson. Ancient microbial communities carry resistance mechanisms that modern medicine did not anticipate. The cave's isolation from antibiotics did not prevent the evolution of antibiotic resistance. It simply preserved pre-existing chemical warfare strategies from before the Pliocene epoch. The less certain but ecologically active cases, brucellosis, Q fever, tularemia, and TBE, operate through warming-enhanced exposure rather than literal resurrection. They are modern diseases in modern animals, becoming more accessible to more people as climate change expands tick ranges, destabilizes rodent cycles, and thaws buried animal disposal sites from the Soviet period. 4 mechanisms drive the risk across all these cases. Preserved biological material in permafrost is the substrate. Exposed burial sites, whether epidemic cemeteries, Soviet veterinary pits, or scattered carcass deposits, are the proximate release mechanism. Shifting vectors, primarily ticks and biting flies, extend geographic exposure beyond the permafrost zone itself. Destabilized animal ecology, including stressed reindeer herds, altered rodent cycles, and new contact zones between wildlife and industrial workers, creates the transmission pathways from environment to human host. Human activity accelerates all 4 mechanisms simultaneously. Roads, pipelines, and mines reach permafrost layers that would not have thawed from surface warming alone for decades. Tusk hunting and scientific excavation open sealed sediments deliberately. Infrastructure development in previously uninhabited terrain brings workers without prior immunity into landscapes where pathogens have been accumulating since the last time anyone lived there. The medical response framework has known components: vaccination programs for anthrax risk reindeer herds, diagnostic capacity for tularemia, and Q fever in remote clinics, tick surveillance protocols for newly affected regions, and carcass safety education for herders and hunters. None of these requires waiting for a crisis to begin implementation. The scientific knowledge to prepare exists. The gaps are in infrastructure, funding, and the institutional attention that remote Arctic populations receive. The scientific lesson from Lechuguilla, Pithovirus, and the permafrost genome studies is that ancient environments contain biology that modern science has barely sampled. The known fraction of permafrost microbial diversity remains small relative to what metagenomic surveys have detected and not yet characterized. The ethical implication which flows from that scientific reality, rather than from any moral argument, is that restraint in reviving ancient organisms in laboratory settings should be calibrated against the actual public health value of the knowledge gained. Pithovirus revival required containment protocols. Reconstructing the 1918 influenza genome required biosafety level 4 conditions and generated ongoing biosecurity debate. Those precedents set the standard. The danger beneath the ice is not only what died there, it is what we forgot was buried.